Okay, I see we already have a few people who joined. So, um, as Martin has a lot of things to say today, I don't want to dally too long. So, I'm going to start with a quick introduction and then pass on the word to Martin. So, a quick introduction about who we are, the people organizing this webinar. So we are Big Omics Analytics. We are a company based in Switzerland, and we developed software for the analysis of omics data. And one of our softwares that is the one that Martin used extensively for the presentation he's going to give today. So Omics Playground is our flagship platform. And with this platform, our goal is really twofold. One is to democratize the analysis of omics data, so make it more accessible to people, who don't really have the bioinformatics or statistical knowledge to perform advanced analysis. And the other purpose is, of course, to reduce the amount of time that people need to get insights from their data by making it more readily available. And I'm sure that a lot of these big questions, why Omics Playground, is going to be answered in full by Martin. You will see he has an amazing presentation. But just to provide you with a quick overview, our platform can really speed up and automate your routine analysis. It provides a lot of plots that you can use in your publications. It's also highly interactive. And it produces, by using best-in-class algorithms and settings, it provides users with robust and particular reproducible results. We also have a lot of databases included for gene set enrichment analysis. We also have 6,000 public data sets, and we are going to further increase that over time. And because it is a cloud-based platform, it's really suitable for collaborative analysis and also easy access to your data 24/7 wherever you are in the world. So before I pass the word to Martin, I just want to give you a quick introduction. So if some of you work in the field of proteomics, you might already be aware of Martin. He is actually quite a leading figure in the field of proteomics. He's the head of the Ghent University Core Facility Unit, the Progenomics Unit. And he has quite an intriguing background, started working with millipedes in East Africa, and then recently moved into what is his main passion, which is decoding the histone code. He has also widened his interest during the COVID-19 pandemic by developing diagnostic approaches that earned him an award with the gained him the UPA Breakthrough Award. And he has quite prominent figures with leading roles in both Belgian and European proteomics associations. So without further ado, let me pass the word to Martin and his excellent presentation.
Thank you, Excel. Welcome everybody. Today I am and thank you, first of all, Big Omics, for inviting me to do this webinar, because I'm very happy to share the latest of our data. I'm gonna talk a bit about how we are switching our omics approaches to focus more on the edges of the biomolecular network rather than on the nodes. because that allows you to kind of find enriched functional associations of the three layers that we're studying, which is proteins, metabolites, and histone PTMs. And the model system we're doing it in is acute myeloid leukemia. So I have to go back quite a while to start this story because it's quite important to have the fundamental biology right. And for that, I go back to the origin of eukaryotes. And that happened when a bacterium and an archaea merged together. And the bacterium became the mitochondria, as you probably all know, in the endosymbiosis hypothesis. And actually, that happened the second time when chloroplasts were engulfed to give rise to the plant. So it's fair to say that bacteria donated energy to the eukaryote experiment. But it was long unknown what the archaea effectively did or contributed. The only thing we do know is that when the eukaryotes started, their genome sizes exploded and they invented multicellularity, which is nothing less but a phase transition in biology. And both of these are two different sides of the same coin of information. And as you might have guessed from the introduction of Axel, I'm strongly convinced that histones played a very strong role in this because that's what histones can do. They can compact a lot of DNA into a very small nucleus, and that's probably why eukaryotes, their genome sizes, as depicted here in distributions, are up to 200,000-fold as big as those of archaea and bacteria. So we have much more DNA. And also, this all needs to be controlled, obviously. So that too happens through histones because PTMs on their tails decide on what parts of the genome become accessible together with DNA methylations, for example. And in that way, we can, from one single cell, an embryonic stem cell, we can start kind of activating different parts of the proteome or genome, depending on what cell lineage the cells are differentiating into. So the multicellularity is differentiation, and that actually means different use of the information that's present in the DNA. And quite recently, people started saying, well, a very appealing hypothesis, I would say, is arising, wherein this interaction between the bacteria and the archaea is still found, is still echoing within the eukaryotes, because it's increasingly accepted that all the metabolites that we find in a cell can actually end up as PTMs on histones. So lactate can become a lactylation, succinate becomes a succinylation. You can go on and on and on. There's new PTMs coming out every year still, up to even serotonin and dopamine that end up on histones as PTMs and mediate, for example, drug addiction, cocaine addiction in rats. So there still is this direct connection between these two. There is a way for the eukaryotes to transfer or to translate the energy of the cell into information so that the cell can adapt itself to whatever challenge is ahead. So that kind of inspired us to build a little new scheme here that we use in-house to facilitate discussions. And it starts from the conventional one, which everybody knows, the dogma, where the genes actually are being transcribed into mRNA. Those become proteins, and they are used to harvest the energy in the metabolites. So this is a linear view that is quite common. But we've added epigenetics to this view by adding DNA methylation and histones. And in doing so, we effectively introduce a pivot point that allows us to fold this line back onto itself, whereby the metabolites become PTMs, which open up new parts of the genome, which allow new proteins to be expressed, and they in turn can then harvest new energy sources, which give you new breakdown products, which can then again be read out as PTM. So this kind of makes more sense in a way because life can adapt very dynamically like this. And we dubbed this the energy information axis because that's what it's doing, right? It's translating the energy into information for the cell. And you could consider this to be a new edge. So here I, for the first time, mentioned the word, the edge. Why edges? Well, we'll define edges throughout the presentation as every correlation between two biomolecules, be it HPTMs, metabolites, proteins, or within the proteins. These are all edges. This is the consistent or coherent behavior of two biomolecules. And I think with this new access, it's a lot easier to understand what I think is generally accepted in cancer biology, that metabolomics or metabolome that is kind of going astray is not a consequence of the tumor. It's a necessary thing to have the tumorigenesis. So with this visual depiction, it's quite clear that that might actually be the case. If your metabolites go crazy, then your epigenetics get messed up, and this way you get different proteins that result in a different phenotype. So this is how we're looking at this. This is what we want to study. And we study it in a cancer model of acute myeloid leukemia, because this one kind of really captures everything we're interested in. It's essentially a cancer of hematopoietic stem cells that stop differentiating and instead start replicating. And depending on how far down the line, you get different kinds of AML. For example, these leukemic stem cells, they are very also phenotyped by a very, very different metabolome, actually. So they have high oxidative phosphorylation, high lipid metabolism, high amino acid metabolism. But there's other kinds of AML where the cells were kind of differentiated to somewhat further stages. And just one very important biomarker I want to pitch here already, because I'll be talking about it quite a bit, is this CD34. Some cells are CD34 positive, others are not. And there is a link with hematopoietic stemness, but it's not one-to-one. So it's not because there's more CD34 that your leukemic cell is more stem-like. So people are kind of figuring out what this means, but it's clinically very relevant and very, very well known within the field. But what makes this AML even more interesting to us is because very recently there was a new therapy introduced that's very successful in a certain subset of AMLs, called menin inhibition. And this menin inhibition is directly targeting the histone PTMs, right? So this is an epi-drug. It works mainly in those AML cells that have this KMT2A fusion, and it affects, or it's a product of many different HPTMs. So that is an obvious reason why we got interested in AML. So we've got the fundamental biology set out, we're looking for the edges, and we're going to use AML to look at them. But to really, just before I'll just first explain how we're going to do this. So what we've done is we had 18 AML cell lines, 6 replicates each, and we sequentially extracted the proteome, the metabolites, and the histones from each cell pallet. So this is just 2 different precipitation steps, leaving us with well over 100 proteome runs, 100 metabolome runs, and 100 histone runs. And all this data we're trying to integrate in 2 different parallel lines. One is the in-house bioinformatics, and I really want to stress that that this will forever be a very important aspect. But also we need big omics here to really tell us in more detail what we see in all these patterns we're finding. And just to show you the in-house clustering that we've done, the UMAPs of the proteome, the metabolome, and the histone here in the bottom, you can see they all have their own kind of separating power, if you will. And if we join them all together, you can see that all the cell lines end up in perfectly separated clusters. So we can argue here that proteins, metabolites, and PTMs effectively add each other or help together to define this molecular phenotype. And it's obviously very rightful to say that whatever these clustering algorithms, especially UMAPs, can give you any clustering you want. Just to show you the first big omics plot, this is the t-SNE plot we got from big omics on the proteome alone. And I also show you this to emphasize just how clean the data is. But you'll be convinced in a minute, I guess. I want to give you one very targeted example to really get you seeing what we're after, right? So there's this one very famous PTM, H3K27 trimethylation, which we and others have found is a very important PTM in stem cell, embryonic stem cell biology. And if we measure it in the cell lines, you can see we can order our cell lines from low to high trimethylation. And I have an arbitrary split here, which I'll come back to later. So we could say there's two kinds of cells, like the lows and the highs. But the thing is, each of these has a different reason why the PTM is either low or high. Because, for example, the first one, Oki AML5, is, when we look it up in a database, has a lot of different mutations in the complex that is writing and erasing this PTM. And these mutations, two of them are even known driver mutations for AML, emphasizing in one go that indeed this trimethylation has a lot of roles to play in cancer. But then the other cells don't have that many. And if you look at the second, third, and fourth cell line, if we go from low to high, you can see how they have this distorted expression in the PRC2 complex. I will come back to that as well. So this is a complex of 4 proteins, you can see here, and they should be together in order to write down this PTM. And you see how the second, third, and fourth one, they have something distorted in the complex. So that might explain why they are in the low population. But then we probably have to move into the metabolites to understand what the high trimethylation cell lines effectively are. For example, in the 6 highest ones, 4 of them have very high methionine, which is the precursor of SAM, the sole donor of methylations to both histones and DNA. So if you don't have a donor for the methylation, you probably won't get much. And then these 2 in the more dark green square here, they have lower methionine, but they also have lower L-glutamine, which through some conversion becomes an inhibitor of demethylase. So that might underlie why these guys have a lot of trimethylation. But you can see this is one molecular target of fewer than 5,000 that I'm just deliberately targeting here to tell you and try to illustrate how these things are so densely connected. And so I'll first have 2 slides to show you our in-house work, and then I'll move into big omics. So what we've done is, with all this data, and this is work from Boris van der Moortelen from the lab of Vanessa Vermeersen, they have kind of clustered all the proteins into modules of proteins that behave in the same way, and that's the blue balls. And then they attribute metabolic and histone PTM regulators to them. And regulators here is a data term. It's essentially just saying that these move in the same or the exactly opposite way. Now, as soon as you have a collection of proteins, you can also attribute a function to that, like with gene set enrichment analysis. So this is what you're seeing here. This is an interactive network. You can see the functions of this protein module. You can then see the PTM, for example, here, H3K27 trimethylation. To what modules is this PTM connecting? And then you can do the same for the green squares, which are the metabolites, like asparagine here. And in this way, you can browse this network. You can do it in an Excel, obviously, if you like that more. But at least it gives you like a connection between these HPTMs and metabolites and these protein modules on the other side. And because of that, we can attribute functions to things that weren't associated to functions before. And this is what I mean by an enriched functional association. So if we, for example, now we're going to see a demo movie of the interface we're building, and it's going to be publicly available whenever it's ready. You can see, you can then just in order to browse that network, you just look for your favorite PTM. It's going to be trimethyl again. You can see what metabolites and what protein modules it's associated to. And then you can look in greater detail to see what the functions of these protein modules are, either or not separately. In this case, you can also just ask, how is my PTM? What is functionally enriched, or what is an enriched functional association to a given PTM? So this is what we define by that. I want to point out this H3K27 is inversely correlating to NADH dehydrogenase activity, and it's positively correlating to a nuclear pore, which we thought was quite surprising, but turns out to be very intuitive. So we've built this, we see the data, we can just have this comprehensive picture, and then we need to make sense of all this. And this is where I want to take you down the rabbit hole, and show you how big omics has allowed us to do this. So trying to understand what all these things are saying. And this is the point where I want to challenge you. If you have a favorite protein that preferably is expressed and known to be expressed in hematopoietic and myeloid cells, just send it in the chat or as a question, and in the Q&A, I'll just see if I can demo a functional interpretation of your protein just live from Big Omics. But to show you how it works, I'm just going to Big Omics. There's no better way. And I'm going to work directly in the interface. So this is how that looks like, right? So it's in the browser. I've got the data view open. You probably recognize this T-SNE plot I showed you earlier with all the cell lines. And what people kind of usually would do is, like I did, I split the cells, these 18 cell lines, into two different groups. The ones with low H3K27 trimethylation and the ones with high K27 trimethylation. So this is an arbitrary and in-house defined contrast, if you will. But if we do that, we do get quite some differential proteins here, which is cool, but that is just what contrasts and volcanoes do. What is really cool is that the third most downregulated protein here is CD34. Remember, this is a very famous AML biomarker. So it's already quite clear that these things are talking to each other, but in what way we wouldn't know. Obviously, CD34 was famous, so we had it as one of the contrasts. You can see me pick here a different contrast. And yeah, if we look at that, we get this conventional thing and a volcano. And often you find your biomarkers and try to go from there. But yeah, so that holds for these as well. But that said, we're doing it on cell lines. I'm not actually aiming for cancer biology here. But if we see, for example, this asparagine synthetase, for those of you in leukemia research, that's actually already a therapeutic target in TALL, another leukemia. So very fascinating that it just pops off a volcano, while I mean it's only cell lines we're looking at. So what people then would do probably is to see if there is kind of a bigger functional enrichment within the contrast. So I'm sticking with the CD34 positive and negative cells here. So this is a known AML target that many people look at. And what people would now usually do is they will do a gene set enrichment analysis. And this means that you order your protein fold changes within that contrast from high to low, from positive to negative. This is the same as walking from here in the volcano all the way down here and then walking back up here. So, and then you look for gene ontology terms that kind of are disproportionately skewed in your distribution. So that's what a gene set analysis, enrichment analysis is. And the first one popping up was not expected. It's branched chain amino acid catabolism. Well, I did mention that these lymphoid stem cells are burning lipids and amino acids like crazy. Well, that's probably what we're seeing here. But especially the second one, well, the fourth in the list, but the others are the same. This decitabine is a known first-line AML therapy in the clinic. Again, we were not hoping to do any clinically relevant work. It's just the first, in this case, second geoterm that pops up. So without really going into this kind of stuff, it's at least really fascinating to see. By now, also people have found more, let's say, elaborate algorithms to find biomarkers, and this is the one implemented in Big Omics. I like it a lot. I'll briefly walk you through it. So again, we're still just trying to split up CD34 positive and negative cells, and all these different cluster algorithms here, XGBoost, Random Forest, you probably heard of a few. They all have tried separately to split your cells according to the metadata, CD34 plus or minus, and they attribute a weight to how well the proteins in the data set are helping the algorithm to do this. So, and then Big Omics kind of brings them together, accumulates all the scores, and then ranks the proteins, saying this is the rank in which the proteins help the algorithm to distinguish CD34 and plus and minus. So then you would expect CD34 to be the first, right? It's not. It's the whatever, 10th or 12th. The order will switch every time you try because it's machine learning and sometimes these algorithms take a different turn. But let's just say there's at least 10 proteins here that seem to have more biomarker capabilities than CD34 itself. Uh, you might think it's counterintuitive, but it's biology, right? So we're expecting noise. There's so many other things going on. And this brings me to really, the core message of this presentation, and that is that these proteins, they're not alone. They are never alone. So if I'm going to the correlation tab here, I just briefly want to point out that there's always different tabs under each tab, but I have no time to go through them all. But so correlation analysis, if you look at how CD34 is expressed in all these different cell lines that are all depicted here, you can see how other proteins behave exactly the same. So irrespectively of whatever, if CD34 goes up in an AML cell line, then this integrin, ITGA6, goes up with it. And it's then no surprise that these two are already well known in the biology field to kind of be co-expressed, and that this integrin helps to attach the cells to the bone marrow. But then like macro H2A, which is a histone variant we're very familiar with, was completely unexpected, as are many of the others, including this ABCC1, which is a chemotherapy resistant gene. So whatever you try, it's going up whenever CD34 is going up. So things are entangled is my core message. And I just explained to you the gene set enrichment analysis and how you can rank all your proteins according to their fold change. Well, we have the same thing going here, but this time they're not on fold change. They are ranked by their Pearson correlation to CD34. So all the way down, you have the anti-correlating proteins, some of which might ring a bell for people that are in the field. And this is exactly what I want to show you in the end, if you have a favorite protein, because what we're doing now is we're no longer looking at cell phenotypes. We're just figuring out how is the biomolecular structure of the network attached. So these are the edges we're talking about. And the thing is, you can do a gene set enrichment on this table. You essentially just download it as a CSV, and then you put it into string. So what then happens here, for example, this is CD34. I've put it in, it takes, let's say, just 30 seconds to 60 seconds, but I just did it before the webinar now. So we've got CD34, and then we have the highest correlating protein. So it's going from here, is positively correlating to the left, the negatively correlating one. So it's a sigmoidal curve. And voila, these are all the proteins, and they are all depicted with their correlation to this one protein. So this is, to me, again, this is and by doing a gene set enrichment on this, you get an enriched functional association. We are not saying CD34 is doing any of the functions here. We are saying that every time CD34 is up, this function seems to be up as well. So it's really reassuring to find that what the marker is famous for, being CD34, is effectively, in at least the wiki pathway gene ontology base, is effectively found. So yes, you get these nice plots here as well. So yeah, you can see the other proteins that are involved in this function. And then, I mean, that these are the things that confirm this is working. But then there's other stuff going on. For example, here, like this is like lipid metabolism, which is positively correlating to CD34 expression. And effectively, I told you that these cells are burning lipids like crazy. But so we can go back to Big Omics and then maybe have a look at that, because we have pathway analysis here. So if I do the pathway analysis in CD34, again, it's the same contrast. The first one to pop up is sphingolipid metabolism. And they have this beautiful interactive plots here where you can see all the like reddish proteins. They're upregulated in CD34. And you can see in blue are all the lipids and the intermediates and how they're converted one into another. So, I mean, from the protein perspective, quite a big chunk of the pathway is covered and it's all upregulated. So this is essentially what we're seeing there in this string analysis. But this now here is very nicely captured through big omics pathway analysis. And if I move to another function I wasn't actually hoping to see or expecting in any way, we see this inverse correlation. So now these proteins are up with positive regulation of chromosome condensation. Now, I didn't know any of these proteins, but you can just click them and it'll give you the function in UniProt. And turns out that these things, they help to condense the chromosomes during mitosis. Now, that just rings another bell in big omics. Hey, they have this really cool function called time series. In time series, if you have a time design in your experiment, so you incubate cells with whatever compound, then this is going to be very straightforward. Like it's the 1 hour, the 6 hour, the 48 hour, whatever, is going to be nicely plotted in this way. We didn't have that, this 18 cell lines. But very cleverly, Big Omics provides you with this extra metadata. And as you can see, it starts with this little dot showing that we didn't make it. Big Omics invented or just, I mean, assessed this metadata. And they just have a set of proteins that are biomarkers for a certain cell phase. And certain samples happen to be a bit more in G1 phase, or I mean, it's all cell cultures. So there is just proportionately more cells in the G1 phase in some samples as opposed to others. And this is what you're seeing here, right? So we plot these three phases, G1, G2M, and S. And yeah, you can just see all the different proteins here. You'll have to ask Antonino if you really want to understand how these things work, but I know it's something with splines. And if you get the very significant interaction Q values here, then these are the proteins that are most affected by the time dimension and so on. This is where it grows above my head, but I always just get support from Big Omics whenever I don't know anymore. Thing is, you can just as well cluster all your proteins here. So this is a kind of k-means clustering. Let's say I want 9 clusters over the cell cycle. And then I see, for example, this cluster 7 here is the one that has a low expression in G2M like my CD34 did. Like, voila, here is CD34. So voila. And then there's some gene ontology on this specific one. I'm not too eager, but voila. So I hope to show now that this is one protein we were pursuing, right? CD34 in a functional enriched environment. But I want to really convince you of this by now going into H3K27 trimethylation. For that, I'm going to look at PRC2. Right, so this is just on the fly. I know EZH2 is a very important one. We've worked on it quite a bit during on embryonic stem cell work. And if we look at what proteins are being co-expressed with EZH2, then the first and second most correlating proteins are two more members of the PRC2 complex. If you remember, I depicted them in the slide, the detailed slide. So this is 4,500 proteins, and the first and second most correlating proteins are effectively part of the same complex. So that kind of tells us that without any wet lab addition in the protocol, we are doing interactomics here. So we are seeing proteins that are physically interacting. That's not what the experimental design is meant to do. It's kind of a really cool feature of the data, which is very rich, which is very noiseless because we've extracted all these different fractions from sequentially from the same cell pellets and so on. And I mean, just when I thought like, okay, then the others probably have something to do with this with this PRC2 as well, I started looking at them one by one, and I was completely blown away when this one, Buzz 1B, we're still in the top 10, was there because with collaborators of ours, we effectively did an interactomics study of PRC2 with the lab from Hendrik Marks and with Dick Zeilmans, and they did a turbo ID. If you're familiar with the technique, I won't explain it, but so they effectively did an interactomic study. It's just out this year, published, and you can see that's the 4 components of the PRC2, and these are all the baits that they got. So these were all attached, and see who's here, like Buzz1B in the plain red here. I didn't know. I mean, I just figured it out, but it's in the data. And there's another one here, MTF2, which I'll come back in a second. So voila. These are incredible things. So again, I know what EZH2 is doing according to the functional databases out there, but now I want to know what is the cellular environment in which this PRC2 is going to be expressed? What will be there whenever EZH2 is there, and what is the functional implication thereof? I just export the CSV, I copy paste it into string, and this is what I get. So here is this embryonic stem cell complex, right? This is the PRC2 as we know it. That's the one I showed you, EZH2, there's SUS12, EAD. That's the MTF2, by the way, that was just recently picked up by the Interactomics study. And then there is the other one I hadn't found yet, but it's clearly a bit lower in the list, RBBP4. And the funniest story of all, we did this interactomics study with the Marks lab because we always see H3K27 trimethylation going the opposite way of H4 N-tail acetylation. So we were actually expecting to fish out an HDAC protein from the interactomics. There is none. We didn't find any. Too bad. So there is in this cluster we didn't find an HDAC. So that's not the story in the end. But it's here. It is correlating in expression. We're not saying it's physically interacting. So if you're doing a turbo ID, you can easily miss out on it. It's just that it's there, right? It's there when EZH2 is there. So that's extremely cool. I'll show you in the concluding slide again that there seems to be an inverse correlation, and I've shown you this in the network analysis earlier as well, that there's an inverse correlation with this NADH dehydrogenase activity. So we can see that again. And there's this vesicle transport. I don't know what to think about or think of this. We'll figure it out in time, but these things are just inversely correlating to H3K27 trimethylation. And that brings me to one very important kind of warning, which again, I was made aware of through Big Omics, and that is that we think in binary comparisons and we define contrasts, but these can be very dangerous. In our case, we effectively also added as metadata the components of the growth medium of our cells. So, for example, certain cells were given this colony stimulating factor, granulocyte and macrophage colony stimulating factor, and others they don't. But if you then look at the fold changes of CD34 positive versus negative, and you compare them to the fold changes of positive growth medium, negative growth medium, you can see that there's a very strong correlation. In other words, CD34 cells, like here's CD34, maybe they're all grown in this medium with the growth factor, but we should be aware that at that point, it is effectively the growth factor maybe that is causing anything. We even have 10% FBS and 20% FBS. Turns out that too has a negative correlation, in this case, negative correlation, but a strong one, to CD34. So this is all things you can just, at one click, you can realize like, oh, I should be careful because these things could be underlying something else. Now, for us, it didn't matter at all because we were after the biomolecular network, not after cancer biology per se. So, but just be aware of this. And then they have a lot of these other really impressive and cool plots like this WGCNA, the whole genome cluster network analysis. I don't have any time to go into this. I'd rather just very, very quickly demo 2 or 3 plots from their multi-omics modules. And again, here, I'm just demoing 1 or 2 plots because you can see how I switched to another data set. This is how easy it goes. Because we are doing omics layers that nobody else is doing. So we have this, we thank Evo from Big Omics to load our histone data into Big Omics. It's not yet a common feature. We will fight for it so that everybody can access in the future. But the thing is, you recognize the exact same things here. But what changed now is that if I go back to the first contrast I showed you in the volcano, now there's also metabolites, and there's histone PTMs in here. Very happy to see that H3K27 trimethylation is upregulated in the said phenotype. But we get also like this hulonolactone. I'm not a metabolomics person, but you get proteins, CDK5, everything is here. I think CD34 is still here, so that's good. Okay, benzyl cinnam is probably the wrong name because that's something with cinnamon. I don't know how these things work in metabolomics, but the features that you're seeing, irrespective of their name, are behaving in this way, and that's what makes it cool. And I want to end demoing Big Omics with this really cool new lasagna plot. You can see how many I have not shown you. They're all there. They're all really cool. But it's just impossible to go through them all in half an hour. But this lasagna plot really caught my attention because this is a layered approach to a simultaneous analysis of genomic and network associations. Really, really long abbreviation for an obvious reason. Once you see it, it's a lasagna. So we have all the different omics layers sitting right here. We can see which ones are talking to which ones by drawing the connections. Voila. And then you see who is influencing who. So this is, again, edges in the network. As you can see, these are correlations that are being plotted. A less cool way of plotting it is in 2D, but it's maybe more practical if it is this way. But you can see where this is going, right? With the purple ones, positive correlations, the brown lines, negative correlations. And this is what they're developing at this point. It's really cool already. It's too bad I don't have more time to walk you guys through this all. But so this was our little rabbit hole. And I want to show you this is where we're currently at by doing one target out of more than 5,000. We targeted H3K27 trimethylation, and you might still recognize the little box plot from all the cell lines. These are the 18 cell lines and their expression of this PTM. And by correlating this expression to proteins and metabolites, we can see that there's a kind of cluster of molecules that always behave the same. And this is where CD34 is sitting. So you can assume that the CD34 positive cells will effectively all have this in their proteomes. You can see here these the lipid metabolism that's upregulated, the branched amino acids, NADH, dehydrogenase, and electron transport chain, this vesicle transport I really briefly mentioned, other PTMs that co-behave and whatnot. So I really want to emphasize here, what I'm showing you is not a cellular phenotype. I'm just showing you what proteins behave consistently. So the question is one goes up, do they all go up? That we do not know. But we do know that within 18 cell lines, they behave consistently the same. These will be the metabolites that will be in the same cell type. Like I said, methionine, by the way, is effectively higher in the H3K27 trimethylation. So, and it all comes back to this Waddington plot. He already knew that cells don't have a free way of just going a bit to the left or a bit to the right. They are stuck to attractive states, to valleys. And these valleys are defined by molecules that always move in the same direction. And this is kind of where we're landing right now. This is the final conclusion. I just want to emphasize that we don't have directionality in the data. It's all correlations. If certain PTMs open up the chromatin and other proteins get expressed. That is the HP axis. But if certain writers of PTMs are going up, then the PTM will go up. And we call this the pH axis. And the same holds here, and the same holds between metabolites and histones. We weren't aware of a histone metabolite axis, but we found one in the data. It's really cool. So this is all coming soon. We will make this data publicly available on the Big Omics website, so you can just cherry pick your favorite targets and get going, deep dive into the rabbit hole. And I mean, people that know me will now go like, yeah, there he is again. This is all framing in a really big like transition in biology, which is driven by this book in part, How Life Works from Philip Ball. And it tells us that we really need to start looking beyond the genome. And at this stage, technically speaking and bioinformatically speaking, we are capable of doing this. We can do multi-omics, we can get them together into networks that make sense, and we can mine public databases to figure out what this all means to us. And with that, I want to thank the whole team at Progentomics. And I want to really thank Big Omics and the people there who have supported us and shown us some extra features in the platform. And so this is my question to you. If anybody has a favorite protein, I think I can just pull it up and hopefully find a few interesting biomarkers. If you've never seen this Big Omics plot to the right before, to the left, you can see a flow cytometry plot, CD34, CD38. This is how people do it in hematology. Well, why not with protein expression? You can simply plot one against the other and you get your samples as if it's a flow cytometry plot. So I think this is really cool stuff. But that's it from my side. I hope you enjoyed it.
Well, Maarten, thank you so much for a really excellent and interesting, stimulating presentation. And yeah, we have some takers on your protein question. So if you can open the platform, I already have two. One is from Xenia, and she asked about HNF1A.
HNF1A. I'm going back to the proteomics
Absolutely.
workflow. I love these cartoons, by the way. Uh,
I'll tell Ivo about that.
They are really spot on. We are interested in, and I hope I have the protein. Obviously, it's only 4,500. So clearly, I cannot guarantee, but so we have H and F. It doesn't
Oh,
seem to work, but
no,
it might not
unfortunately.
be the right name.
Hmm.
HNF1A. Is it? No, that's exactly how it's called. So I'm sorry, it's not in the data. Somebody
And
else.
I do have two more. So there is one from Omar, which is ADGRE2. Ah, no, we don't
It's the
have it.
five. It's the five version. It's a very similar one.
Okay,
We
okay.
can have a look and see it's an adhesion G protein coupled receptor E5. So it's coupled to these transmembrane proteins, which I think makes sense, no? Especially the lipoprotein receptor that is attaching here, integral membrane. So it's pretty obvious. Adhesion, all the other ones are also adhering the cells to something clearly. So I don't know these proteins myself, but you know what? Um, I'll just look into the like enriched analysis in string later on, and I'll send Omar the result, the link. Then he can have a look himself. But probably Omar, you see some proteins that make a ton of sense because they all seem to be like, yeah, attached to the membrane somehow.
Yeah, thank you very much. I'm excited that I can see CD37 up there, actually. Just
Cool.
want to say hi and thank you.
And there is another protein that's from, I hope I'm not mangling your name. Forgive me if I am. There is Aka Emmanuel, and it's IGF BPL1.
IGF 2BP.
BPL. I don't
Yeah,
think it's there.
it's not there. It's another one. I don't know if this one even is remotely related. Sometimes they are if they have a similar name. Is it insulin-like growth factor we were after? I don't know. But so yeah, that's I mean, obviously this is also where we're applying for a new machine. Everything here was acquired on a SciX Xenotoph. It's a really good instrument. But we are sure with every next generation we get more proteins out, and if we have 7,000 here, that just increases the chances of us finding all these interesting targets. But yeah, thanks for the challenge, guys.
Yeah, if there are any other questions from our users, you can even activate your microphone and ask them directly. You don't need to type them. So feel free to ask. Maybe raise your hand first, and then we will call you out. Okay, I don't think I see any. But well, if you think about them, I actually, I really liked the use you made of the platform, and in ways that I don't, I'm usually the one that uses the platform most, and I really like the way that you made use of modules that I normally don't spend a lot of time on to find new and interesting correlations. And I'm thinking, how, what sort of, I see, for example, you were including the string data, to then play between the platform and the string database. Do you think, for example, there is something that we could do to make this interaction more flowing between the two? Maybe even have some ways to do that directly, from the platform, rather than you going to a separate website.
Yes, if there would be like a direct button, submit to string, that would be a really cool feature because string, whether you like it or not, has a few really cool features that are for the large part embedded already in big omics. So this, I might have misled people by thinking that it's a different place. No, so there's most of that can be found in big omics, but they explicitly map the proteins on your protein distribution. And you can see, I mean, a number of an FDR or something is a number, right? But if you see how there's like 5 proteins dead on in the same place in your distribution, you feel like, okay, these are together. So I like deep understanding of the numbers that are given to me. So yeah, I think a direct export button from that correlation place would be a very nice one.
Oh, that sounds great. And Antonino is one of our main developers on the call, so I'm sure he has taken note of these interesting changes. I think there is a question from Omar.
Okay.
So Omar, please feel free to ask.
Hi, Martin. I have a quick question. Yeah. Hi.
Hi.
So I just wanted to know your input about the feasibility of using phosphoproteomics as an extra layer to define the cell function here, or if there is a kind of analysis, or just looking at the data in a holistic way, because sometimes you might have the protein or the gene out there, right? But the pathway is not really activated, especially like in a very complex disease like leukemia. Just want your
Yeah.
input.
Thank you very much for the question. It makes a ton of sense. Yeah, so this is in other presentations that are more about the biology. I emphasize the limit of sticking to abundance only. Abundance has a correlation to activity, but it's far from one to one. It's more in the ballpark of mRNA protein correlation. So abundance is just like if there's a lot of a protein there, it's probably it has a higher chance of doing something. But effectively, so many proteins they need a PTM to be active, and this is kind of also the if I'm maybe I quickly just reshare if I find the button again. Yes, here I am. This is what I had in the conclusion. So in this little slide here, if you're seeing it again.
Yes.
Yes, so this is exactly what you're saying, right? So some metabolites, they will activate or inactivate the proteins, and then you can have as much of the protein as you like. It's not going to change anything. It's effectively the same thing as what I showed you here, where a mutation will make sure that whatever amount of protein you have, because the first cell line here has quite a bit of this PRC2, right? But it's all mutated. So you can detect it with a mass spec and you think you have a lot of it, but it's just unfunctional gibberish. So you're so right that we are limited by looking only at abundances, and including phosphoproteomics in this would be a major addition. Then we could say we have so much of the protein and we have so much phosphorus on that protein, implying it's active in this way. So, but that is, I think, a big headache for big omics, because getting that in will be a very tough one.
Yeah. No, thanks for acknowledging that for sure. I just was just curious to know if there might be any computational way to infer that, just like, thinking out loud, because experimentally speaking, I know that phosphoproteomics is kind of finicky, you know, to
Yeah, it
produce
is.
accurately. Yeah.
It
Thank
is.
you.
Thank
Appreciate it.
you.
Uh, if there are any other questions, otherwise, I think we can wrap this webinar up. And I would like really to thank you, Maarten, for not only an excellent presentations, but also providing us with some interesting ideas. As always, your feedback on the platform is really spurring us to keep improving our platform. And it's a bit what we try to do. We really act on our users' feedback. Especially when it's pretty obvious that it's something that is needed by the community. And as Maarten said, his data sets will be made available on our platform. And for those interested, I'll quickly share. Let me see if I can find it. Should be here. Yeah. Just a link where you can both ask more information, but also you can sign up for a free trial version of the platform. And in the future, we will make Maarten's data set available through the free trial version of the platform. So you will be able to play with it just like Maarten did on his presentation. So I'll think we can finish off here. Oh, there is a message on the chat. Let me just check. Okay, yes, it's just thank you for the compliments, guys. As I said, thank you so much to everyone for attending. We have actually made a recording of this session, and we will also make it publicly available as once we have edited it. So thank you so much, everyone, for attending. Thank you so much, Maarten, for
Thank you,
as
guys.
always, a very stimulating presentation.
Thank you. Goodbye.